Hell.
Transporter proteins. Hey there, welcome back to our channel. Today, we're diving into the fascinating world of transporter proteins. If you've ever wondered how substances move in and out of cells, you're in the right place. Let's break down the different types of transporter proteins and their roles in our bodies. First off, what are transporter proteins? Think of them as the gatekeepers of our cells. They help move molecules across the cell membrane, either bringing nutrients in or getting rid of waste. There are two main types of transport, active and passive. Active transport requires energy, usually from ATP, to move substances against their concentration gradient, meaning from an area of lower concentration to one of higher concentration. This is like swimming upstream. On the other hand, passive transport lets molecules flow down their concentration gradient without using energy, similar to a leaf floating down a stream. Now, let's focus on active transport, which can be categorized into two types: primary and secondary active transport. Primary active transporters use the energy derived from ATP hydrolysis to move substances. A classic example of this is the sodium-potassium pump, or Na+/K+ ADPase. This pump moves 3 sodium ions out of the cell and 2 potassium ions in, helping maintain the cell's membrane potential. It's like a battery that keeps our cells charged. Next up is secondary active transport. This type doesn't directly use ATP. Instead, it relies on the electrochemical gradients established by primary active transporters. For example, the sodium glucose transporter uses the sodium gradient created by the Na+/K+ pump to bring glucose into the cell. It's like hitching a ride on a bus that's already moving. Now, let's look at the various types of transporter proteins. 1. P-type ATPases. These are primary active transporters that get phosphorylated during the transport process. They're crucial for maintaining ion gradients. 2. V-type ATPases. These transport protons into organelles, helping maintain pH levels inside cells. 3. F-type ATPases. Found in mitochondria, these transport protons to generate ATP during cellular respiration. 4. ABC transporters. This is a large family of transporters that move various substrates, including drugs and lipids, across membranes. They play a significant role in drug resistance, especially in cancer therapy. And let's not forget about nuclear transporters. These proteins are essential for moving macromolecules like proteins and RNA in and out of the nucleus. Importins and exportins are key players here, ensuring that the right molecules get to the right place at the right time. Importins bring cargo into the nucleus, while exportins help move materials out. This process is vital for gene expression and cellular function. Transporter proteins are essential for maintaining cellular homeostasis, allowing cells to take in nutrients, expel waste, and communicate with their environment. Understanding these proteins not only helps us grasp basic biology but also sheds light on how drugs work and how diseases can disrupt these processes. How about nuclear protein transporters? Now, you might be wondering, what exactly are nuclear protein transporters? Well, let's break it down together. Nuclear protein transporters are essential proteins that help move other proteins and molecules into and out of the nucleus of a cell. You see, the nucleus is like the control center of the cell, housing our DNA and regulating gene expression. So, it's super important that the right proteins get in and out efficiently. Transport happens through structures called nuclear pore complexes, or NPCs for short. These NPCs are like gateways in the nuclear envelope, which is the double membrane surrounding the nucleus. Think of them as security checkpoints that only allow certain cargo to pass through. Now, let's talk about the key players in this transport game, karyophrins. These are a specific type of transport receptor, and they come in two main types, importins and exportins. Importins are responsible for bringing proteins into the nucleus, while exportins do the opposite, sending proteins out to the cytoplasm. Here's how it works: importins bind to their cargo in the cytoplasm. Once they reach the nucleus, they encounter a molecule called RANGTP. This molecule has a stronger binding affinity than the cargo, which means it can kick the cargo off the important and release it inside the nucleus. Isn't that cool? On the flip side, exportins bind to their cargo in the nucleus and also to RAN-GTP. Together, they form a complex that can travel through the NPC to the cytoplasm. Once they're outside, the complex disassembles, releasing the cargo back into the cytoplasm. Simple yet effective. But it's not all smooth sailing. Dysregulation of these transporters can lead to serious problems. For instance, improper nuclear transport is linked to diseases like cancer and neurodegenerative disorders. It's like having a faulty security system that lets the wrong things in or keeps the right things out. One interesting example is how the HIV virus uses a transporter called transportin 3 to sneak into the nucleus and replicate. And in some cancers, mutations can disrupt the transport of proteins like cyclin D1, which can promote tumor growth. But what happens when these transporters don't function properly? Well, that's where things can get a bit tricky. Dysregulation of protein transporters has been linked to several diseases, including cancers and neurodegenerative disorders. Let's start with cancer. For instance, the XPO5 transporter is crucial for exporting certain RNA molecules out of the nucleus. When XPO5 is dysregulated, it can lead to impaired microRNA maturation. This has been observed in colorectal cancer, where it promotes the expression of oncogenic microRNAs, basically, those that can drive cancer development. Next up, we have transportin 3, or TNPO3. This transporter is particularly interesting because it binds cargoes that contain specific sequences, helping them enter the nucleus. Interestingly, the HIV virus exploits TNPO3 to facilitate its own nuclear import and replication. But mutations in TNPO3 have been associated with limb-girdle muscular dystrophy, and surprisingly, they also provide strong resistance against HIV-1 infection. Now, let's talk about Alzheimer's disease. Abnormal expression of ATP-binding cassette, ABC, transporters in the blood-brain barrier can lead to serious consequences. For example, the accumulation of amyloid beta, a hallmark of Alzheimer's, can occur when these transporters fail to clear neurotoxic molecules from the brain. This disruption can contribute to the degeneration of brain cells and the progression of the disease. And speaking of ABC transporters, ABCA1 is another key player in the brain. It helps regulate cholesterol levels, which are crucial for brain health. Altered cholesterol metabolism is linked to an increased risk of Alzheimer's, especially in individuals carrying the ApoE4 allele. This allele is known to promote the aggregation of amyloid beta and plaque formation. So, as you can see, the role of protein transporters goes beyond just moving molecules. They are vital for maintaining cellular health. When they malfunction, it can lead to a cascade of problems, ultimately resulting in serious diseases. To wrap things up, understanding how these transporters work and their implications in diseases can pave the way for new therapeutic strategies. Researchers are actively looking into ways to target these transporters to improve drug delivery and combat diseases. Thanks for tuning in today. If you found this video informative, don't forget to hit that like button, subscribe for more content, and share it with your friends. Until next time, keep questioning and exploring the wonders of science.
PharmaPal.